Good afternoon, everyone. I'm Ivan Ponomarev, Manager of Chemistry and Technology Department at Elmarco. Today we open Elmarco webinar series, which will be focused on applications and electrospinning process based on needleless and nanospider technology. And I would like to welcome today our guest from Technical University of Liberec, Associated Professor Eva Kuželová Košťáková and Professor David Lukáš. So we are sitting here together in Czech Republic at Elmarco company. And now I want to turn the floor over to Katarzyna Rubáčková, our Master of Laboratory Equipment from our Customer Support and Sales Department. And she will make a quick introduction of our technology, mainly laboratory equipment.
Yeah, thank you. It's my great pleasure to share with you a couple of slides introducing Elmarco company. First of all, Elmarco is basically a global leader of supplying production scale technology for making nanofibers. We are a global company existing for more than 20 years in the market and having over 250 installations globally. Our technology is named Nanospider, is the brand name of the technology, and I want to describe it a little bit more in details. The core of the technology is a wire. So basically, the wire is working as a spinning electrode where the polymeric solution is deposited on such a wire, and spinning is taking place from the surface of the wire towards the collector. The collector is basically also the wire, but we are depositing on the moving substrate, which can be typically any kind of non-woven textile substrate, could be paper or some kind of foil. By actually correlating the speed of the substrate, we can control the thickness of a deposited membrane, which is leading us to different possibilities of applications. You may also follow the video, which is demonstrating the real process which is taking place in the spinning chamber. So the portfolio of Elmarco is, I would say, broad, because we are delivering not only the industrial scale, which is actually forming our core business, but we also supplying the market with the laboratory research equipment, which is the starting tool for all researchers. And the biggest advantage, I would say, is the possibility of upscaling. So whatever is developed on a small scale lab equipment can be easily transferred to the industrial scale. Now I'm coming to a variety of applications which are actually where nanofibers are benefiting. There are several segments in the market that take advantage of nanofiber properties. Some of them are fully developed. You can meet already the commercial products made out of nanofibers, especially in the air filtration segment. But there are other segments which are rapidly growing at this moment, and one of them is as a medicine. And medicine is actually the main topic of today. Medicine is also covering different sub-subjects. And today our main speaker will be focused on wound care and tissue engineering. Before I give the room to our main speakers, I'd like to invite you to the Medica exhibition, which is taking place in 2 weeks in Germany, Düsseldorf. So whoever has a chance to visit us, we are in hall number 3, booth number 8. So come there, because we're going to have a machine there. People from Technical University is going to be there with the product. So it's going to be a great opportunity for you to discuss the applications. And whoever wants to have a free tickets, also email us, because we have some free tickets for you to be used. So thank you for attention, and now back to webinars.
Thank you, Katarzyna. Perfect presentation. So I would like to further present our guests, Associated Professor Eva Kuželová. She's a member of Bioengineering Department of Chemistry, Faculty of Science, Humanities and Education at the Technical University of Liberec. She is primarily devoted to material engineering associated with the creation of fibrous materials for medical applications and their testing. Our second guest is Professor David Lukáš. He's the head of the same department, and he's a leading expert on the physical principles of the formation of polymer nanofibers, the author of many patents and the author of important publications in the field of electrospinning, both types of electrospinning, direct current and alternating current. So if you have any questions, please write down or use the Q&A section of this webinar. And now, please, you can continue with your presentation.
Thank you very much, Ivan. Thank you very much. So, first of all, I would like to thank Elmarco for the possibility to present here as presenters for the medical applications of nanospiders. So here we are, and here is our presentation. So at the first, I would like to introduce that we at our group of bioengineering are focusing mainly to the teaching. We guarantee the bachelor and master study in the field of bioengineering, but we also do, of course, the research mainly in the production of polymeric nanofibers by all kinds, or not all kinds, but many kinds of the spinning technologies. And what we are really proud of is that we are also able to do in vitro testing, the biological testing of our mainly nanofibrous, polymeric nanofibrous products for the medicine. We are able to test its cytotoxicity, viability, proliferation, migration, hemocompatibility, or other processes connected with the biodegradable nanofibrous materials, as the testing of the degradation is. Here I have on this slide some typical pictures of the cells which are seeded on the nanofibrous, electrospun nanofibrous material, or also the nanofibrous material with the red blood cells, or the degraded nanofibrous material. So you can see how fascinating our world is. What we today would like to tell you, we would like to introduce not only needleless electrospinning from static wire, but also other possible technologies for the production of nanofibrous materials for the medical applications. We would like to tell you why we think the nanofibrous materials are for the medical application really unique, for which applications we chose as the best, really, the DC electrospinning. It means the nanospider technology. I will show you several studies, the new yeast published last year or this year, and we will finish with the description of the clinical study, which we are really proud of. So I usually start my lecture when I have it in the first year of the first semester, the first lecture, with the article of the Professor Whiteside from the Harvard University. And in this article, it is about the bioengineering. It is about the combination of material science and the biology and the medicine. And Professor Whiteside had many claims there, but I would like to introduce here three of them, because I think that they are extremely important for us. At the first, material science, it means the materials engineers who produced materials for the medical application. We have to follow and serve. We aren't leaders. Unfortunately, the leaders, we aren't. Leaders are the medical doctors, surgeons, and so on, who has to define the problems, and we have to follow and serve. Other thing which we still have to think about is that all the medical problems are like a puzzle, and the production of material is only one piece of the puzzle. And the last claim, which I really love at all, it is that who start to be bioengineer, who would like to produce materials for medical application, will have endless fun. So let's have a fun and produce materials for the medicine. So now I would like to shortly, briefly introduce the technologies which we can use for the production of polymeric nanofibers for the medical applications. I wouldn't like to say that there is only one technology all over the world. This is why I have here this overview. So first of all, here is the DC electrospinning, the technology which is, I think, the most known all over the world. If it is needleless, with different kinds of electrodes, spinning electrodes or collectors, etc., etc. Beside the DC electrospinning, we have here the AC, it means alternating current electrospinning. And we have to say that both of these technologies use the electrical force for the transforming of the droplets of the polymeric liquids, which we used, mainly this is polymeric solution or melt, to the form of the fibers. If we don't want to use electric force, we can use centrifugal force, or we can use the hydrodynamic force for the solution or melt blowing. We can also use the mechanical force for the drawing of individual fibers, etc., etc. We have many other possibilities, as we have a shear spinning, flash spinning, laser spinning, and so on. There is a number of different technologies. If we focus to DC electrospinning, because today we are here because of the DC electrospinning, we have to say that we have really versatile technology, which can offer us many possibilities. We can change many of the material conditions and also process conditions. And we can produce really a huge amount of different morphologies or the structure of the nanofibers. The truth is that compared, if we would like to compare needle and needleless electrospinning, we have to say that, for example, coaxial nanofibers, hollow fibers, or side-by-side fibers are mainly produced by needle electrospinning. But there is plenty of materials which we can produce by needleless electrospinning. Also, of course, a representative of the needleless electrospinning is the nanospider technology, where we are using the static wire electrode. And you can see that what I would like to underline is that mainly if you would like to produce membrane, two-dimensional layer, it is extremely good to use needleless electrospinning, especially the nanospider. But you can see that there can be classical, isotropical, random-oriented fibers, porous fibers, beaded fibers, tapes, also the composite nanofibers, etc. You can, with the different properties, mainly the process properties, also created nanonets in between the nanofibers, or you can produce pattern nanofibrous material. But as I said, I wouldn't like to tell you that only the DC electrospinning is here. So if you would like to produce some bulky material or linear material as yarns or threads, there is possible, or maybe better for some applications, even for the medicine, where you would like to produce surgical sutures or the dental flosses and so on, to use AC electrospinning. You can also use the solution blowing, or sometimes the technology is called air-jet spinning, if you would like to exactly apply your nanofibrous material to the place where it will work, so-called in situ application, which is possible here because the solution blowing doesn't use the electrical force. And the last technology, which I would like just briefly introduce, is drawing, where you are able to produce individual fibers or bundles of fibers. And these materials are used for very, very special applications, as for example, following spinal cord injury regeneration. So back to the needleless electrospinning from wire, let's say the static wire electrode. Why we chose these technologies for the production of our materials? Mainly when we would like to produce membranes, as for example wound dressing. We really have to work with some productivity, because we wouldn't like to spend all days with the needle electrospinning. So this is why we think that it's wonderful to use the nanospider. Other very positive thing is that you can start really the scaling, as Katerina already said, that you can start from very small amount of the solution, let's say units of milliliters of the solution, and you can set up all the process, because for the medical applications, usually the solutions, polymers, additives are extremely expensive. And once if it is set up, you can spill, let's say, from liters. And it means that it's wonderful, and not all the devices offer this possibility. Other thing which is, according to me, extremely important is the repeatability for the medical applications. Because if you once produce something and in 1 month or in 1 year, you have to produce the same thing, because it's extremely important here. And what is important is still to control the conditions during the electrospinning, and the climatization unit, which is here, nanospider as a part of the nanospider technology, let's say, is extremely important. In last years, we made many studies where we really proved that mainly the humidity, air humidity in ambient conditions is extremely important. Also, there is important the uniformity in the layer, which cannot be so good, or which isn't so good for the needle electrospinning. So now I would like to briefly show you some studies which we did in last years. So firstly, and all materials which we presented here in these studies were produced by Nanospider. So first of them are electrospun biodegradable materials from polycaprolactone. It's a biodegradable polymer, and we were tested them with the integrated antibiotics, lipofosphanoxine, which are very interesting because our body doesn't induce the resistivity for them. So they are good for wound dressing, and also not only for the acute wounds, but also for the chronic or burns. So we made a wonderful study with our colleague from Institute of Organic Chemistry and Biochemistry, Academy of Sciences of the Czech Republic, where we showed also in vivo testing, and in mice, it has really wonderful results. Other two studies, because I'll continue, are focused to the electrospinning of polyvinyl alcohol nanofibers. It means water-soluble nanofibers. The water-soluble solution, or water-based solutions, offers us the possibility to incorporate it also active or sensitive agents, as for example, proteins, or in this case, it was the platelet lysates are. And here we were really successful also in a study with the treatment or healing of the chronic wounds, mainly for the diabetics. The following study has the same core material, or the same raw material, let's say, just the fibers were deep coated into the fibrin material. And finally, we were able to increase the proliferation, migration of materials. And also it was wonderful how keratinocytes were able differentiated. I'm sorry. Also, of course, these materials can be used as antibacterial layers, what is also important for the wound dressings, but not only for them. And we will be in part of the groups who dealing with very fascinating topics, how the morphological properties, let's say, of the materials can influence the growing of the biofilms of, for example, Staphylococcus aureus. And the last study which I would like to introduce to you isn't exactly the material for the direct application, like a scaffold for tissue engineering or for drug delivery, but it is a material which we produced again with nanospider, and it is material which finally in fibrous form contains 95% of nanoparticles, and only 5% is a polymer there. So it looks a little bit like a stocking full of stones, but still nanospider is able to electrospin it. It works there. I was really surprised how it is, and for us, it was mainly for X-ray shielding, for the protective clothing, mainly for the medical doctors or surgeons who are working under the X-ray. So that's all, and now Professor will continue.
Thank you for the floor. I would like to begin with something what relates a little bit what Eva said. It seems as that everything is smooth in the case of the application of nanofibrous material to the realm of medicine. On the other hand, there is something like a contradiction. Electrospun nanofibrous material really have a very big potential to be applied for medical application like wound healing materials or tissue engineering materials. On the other hand, despite this potential, there is no recent impact of this material to clinic. It is not my statement. It was announced, but Stoddard in the year 2016, and that is why there is really a question why the recent state is so. It should be thanks to the question if nanofibrous material are capable of overcoming the biological safety and efficiency challenges, or if electrospinning process is not matured for such applications. We would like to tell you our answer to this challenge. First of all, I would like to highlight once more that I am proud that Technical University invention was in fact something like the beginning of Elmarco entrance of the global market with nanospiders. Our first invention was to base these machines on so-called cylinder spinning electrode. On the other hand, I would like to underline that recent spinning electrode that Katerina described to you, it means the string or something like a wire, is really a spinning electrode that has a lot of advantages. I would like to continue really with the application of nanofibrous materials to the area of wound healing. First of all, I would like to underline that nanofibrous materials, artificial nanofibrous materials that are on the left of the screen, are really very, very morphologically similar to the extracellular matrix of our skin, as you can see on the right-hand side. Why the time is ripe to the application of these materials to medical field is, for instance, something like a citation report. This one is done on two keywords, nanofiber and wound. So it is visible that back 10 years, it started a lot of articles that are dealing with this topic. We can also why it doesn't work. Yeah. Fine, thank you. We can also to mention the former development of the market with nanofibrous products worldwide that are devoted to medical application. That's visible on this graph, and also there is something like a future prediction of the global market with the nanofibers for medical application. And you can see that we anticipate very important or very significant growth of it between 2021 and 2025. Now I would like to tell you something about the case study of the material that has a brand name, Nanotardis. It is an electrospun nanofibrous material for wound healing. It is made by polycaprolactone, and we carried out successfully clinical trial with this material. And I would like to underline that samples were produced by Nanospider. We started in 2014 with something like the casuistic. Here you can see the material, how it was applied to wound. It is just a poor polycaprolactone nanofibrous layer. Here it is after the sterilization, and here it is with some information for the usage. The first casuistic was done with the illness, with the cancer illness on the head. Here is the beginning stage or the original stage of this illness after the excision and after the healing. After the first casuistics, we jump to really the clinical trial, and we also formulated something like the objectives for it. The first of them was to show the suitability of nanofibrous materials for such usage. This material is biodegradable, and that is why it was not necessary to remove it from the wound. And then also something like a goal was to show that this material heals thanks to its morphological properties, because it resembles the structure of extracellular matrix. The hypothesis of the clinical trial was based on this statement, and also we anticipated that this material is permeable for airborne oxygen, and also it should be used as antibacterial barrier, what is also profitable for these materials. Here is an evidence that we received the decision of the SUKL, what is the Czech equivalent of the American FDA, to carry out this clinical trial. Here is an evidence of the final report, unfortunately written in Czech. And I will follow with some information about this clinical trial. Here is, for instance, the cumulative number of the subjects against the date. You can see that we were tested around 20 subjects. Here is something like the demography of these subjects, the date of birth against the number of subjects. And here is the division between chronic and also clean acute wounds, and also the distribution of the localization of the lesion findings. Now I would like to tell you something about the analysis that is related to this clinical trial. First of all, our quantitative data was based on the measurement of the biggest and smallest radius of the wound. It is called ferret maximum or minimum ferret diameter. Here you can see the dynamics of the healing, the original state, the state after nanofibrous material was applied, and the healing process. The main advantage of nanofiber material is thanks to the fact that the scar is really not so huge, as the healing process ends without nanofibrous material. And here is an experiment that shows you that nanofibrous material in the wet wound doesn't need any binding. So that is why by capillary forces it is incorporated into the wound. So that is why it is really something very gentle to treat subjects with this material. Here are some information about the dynamics of the healing process. For instance, for a dozen of subjects, you can see how the maximum Farad diameter changes during the days of monitoring. And here we try to collect all the data to show that we are able to predict the, more or less predict the healing process. On the horizontal axis is something like dimensional time for healing. It means T0 is the final healing time, and here is also the dimensionless of the diameter of the wound that is divided by the original dimension. And now I would like to show you on this example that was provided by our friend, by Primář Zan, that nanofibrous material was applied to old wound that was covered by so-called or healed by so-called skin graft. You can see that in this area, the healing was something like, or the result was something like a wax-like skin. On the other hand, here it is also a detailed photograph of it. Here is the skin graft, and here is application of nanotardis, and you can see angiogenesis strongly developed in this area. So that is why the final healing process is more resembled natural skin. Here is something like an evidence that our clinical trial is just now registered by EUDAMED databases. It is the European databases of these trials. So thank you for your attention.
Thank you.
So thank you very much for a very interesting presentation, very informative, I think, for all of us, definitely. So I see that I just want to remember that you can write your questions to Q&A section, but I think I have to use my priority and moderating position of this presentation today, and I want to ask you the first question. And the first question is actually, you showed a nanotardis material. What is the effect of thickness and basis weight of this material to the healing performance?
So first of all, I would like to answer the question about the mass per square meter. It was between 15 and 30 grams per square meter. And the healing process was started with scissoring the same shape as the wound has. And for these clear wounds, it was applied just once. And the nanofibrous material either disappears in the wound or it was reacted by it.
Okay. And is there any thank you, and is there any correlation between healing performance and the basis weight, the grams per square meter?
Like a thickness?
Yeah, like a thickness.
So as David already said, the material almost disappeared, and we are absolutely not able later tested it. So it won't be nice to ask the patient, okay, now your wound is healing, can I take a piece of it and test it at the histology, for example? So, and all over the world or in the literature, there is only a less about this, what you ask, because it's excellent question. But what we can say that we have these 30 grams per square meter and 15 grams per square meter, but mainly because of, let's say, drape. Because if it was for the children, they really need to have the lower, let's say, mass per square meter, because it has to be more drape, more sensitive on their wound. And on the other hand, the material has to be still handled. for the surgeons. They have to work with it. So this is why they wanted to have a little bit higher mass per square meter. This is what I said, that sometimes you think that you produce wonderful material, that it will work. And finally, the medical doctors, they said you, but it is bad because I cannot cut it, or I cannot take it in the gloves, for example, like this. So still in puzzles, but the truth is that we haven't right answer how it is with the thickness of the material and the time of the healing. We just use these two because the surgeons said that it would be the best.
Okay. Thank you very much. Very good and complex answer, I have to say. And I'm feeling electrostatic field from Katarzyna. She is obviously willing to ask another question.
Yeah, I'm seeing there are a lot of questions coming through chat, so we definitely going back to that. But I have a question to Eva about the sterilization of these materials, because I'm getting a lot of questions from our customers who are buying the equipment. They want to place them in a clean room, very high classes, to protect it and to really produce it. But I know your equipment is not really placed in any clean room. So how do you deal, let's say, with the to be worked out for the medical application, because I believe it needs to be sterilized in some point. So how this is done?
So definitely sterilization is extremely important other piece of the puzzle. So it works just now for me that I spoke about it. And I would like to say that yes, you are right. Our machine isn't in an absolutely clean room, but it is enough for the testing in vitro, in vivo, and the clinical study. Of course, it a little bit differs for the, let's say, necessities if you are really the producer. Each producer has to then follow the laws in their country and so on. We are producing the materials, then we pack them to the special, let's say, bags for the materials for medicine, and we sterilize them. It is really a huge task because, as we spoke, for example, about nanotardis, it is made from polycaprolactone, and I think that most of you know the polycaprolactone has a melting point at 60 degrees of Celsius. So there is not possible boiling, steam, and so on. And we are mainly using sterilization by ethylene oxide, by gas, and by the gamma irradiation. Both of these sterilization techniques are possible for human use. We don't want to use sterilization, for example, by UV and so on, because these aren't certificated or aren't able to be used for human clinical trials, but also for the clinical application. So both of these can be done at the room temperature, what is excellent for us. And we have also the ethylene oxide sterilization exactly at our department. So it's as easy as it is. But for the clinical trial, it was necessary to sterilize our material in a certificated laboratory. So we asked the laboratory in a huge hospital in Prague who are able to give us that certificate.
So there is, in other words, saying there are opportunities to find like certified laboratory in Czech Republic, let's say in some hospitals.
Yes, definitely. Or for the gamma irradiation, it's a special company who is dealing with the gamma irradiation for the sterilization, all kinds of materials. So it is definitely possible. Just we have mainly experience with the production of materials for in vitro, in vivo testing and the clinical trial. We aren't producers, so we cannot answer this question.
And I would expect that with the gamma radiation, you could sterilize the material even in a bulk, meaning in a roll.
Definitely,
So
and also the dose is important there. We can ask which dose we would like to have, for example, 15 kilograms or something like that.
Great, thank you. So let's move forward to the Q&A section. The first question is, you showed a lot of beautiful defects in nanofiber morphology, something like pores, shish kebab beads or flat fibers. What do you think? Is it really possible to let's say control the formation of such defects, which sometimes could be beneficial?
If I can answer, so we thought that most of these aren't defects, but we wanted them there. For example, the beaded materials can help to increase the hydrophobicity if the material originally is hydrophobic. If we produce the, let's say, in the post-process treatment, the shish kebab structure, we hope that higher surface can help to adsorption of cells and so on. And your question is right. This is why I said that we really like nanospider for these that we can start with small amount of the solution, really set up not only process parameters but also the materials one, because these are extremely important. How we prepare the solution is important if there will be finally beads or if we will have tapes and so on. And the truth is that if the setup process is done well, let's say it's well done, then it is really uniform over the layer. So it is definitely possible. And all of these images which I showed there were produced by Nanospider. Of course, I am not able just now to give you a proof that in all places of the complete membrane it was the same. But believe me that if you set up right the material and process conditions, in this case with Nanospider, it's definitely possible to produce it with the uniformity in the membrane and with the repeatability in time.
Thank you. Thank you. So moving to another question, I think this question will be for more Katarzyna, maybe even. What would happen if you use a positive high voltage in the wire and negative in the collector side, for example, +20 and -20 kilovolts? Is it different compared to +40 kilovolts and zero potential on the collector?
Yeah, I would like to say that this 20 kilovolts is for our technology not much, right? That most of the processes are starting to span at maybe 50 and higher kilovolts. So, but okay, I understand the principle or the point of your question. So, for example, if you have a positive high voltage, let's say 60, and on a collect and a negative, we have 20. Altogether, you generate 80. You generate, of course, more power to the system. And your question was if there was 60 and for example 0 on the top, right?
Yeah, meaning that the sum of potentials, is there any difference between sum of potentials or just one bigger potential on one or another electrode?
Yeah, I would say that, yeah, I mean, we take it as an entire, let's say, sum of potential or the voltages. So we take it as a total number, comparing 60 kilovolts to 80 kilovolts. And I don't personally notice big difference if there is a 0 or on the top, but maybe Professor has
some ideas.
So I would like, you are completely right, but I would like to add, if the collector is on 0 potential, then any part of your device is also on the 0 potential. So that is why then there is a problem to focus fully your nanofibers on the substrate. So that is why, in my opinion, I will recommend to use minus something on the collector.
Just I would like to say that theoretically there is no difference, because the important is difference of the electrical potentials. So as was the question, +20, -20 or 40 and 0. Theoretically there is no difference. From the practical point of view, David already said the right answer. And I think that mainly these 2 high voltage sources are there because if you really would like to be high and you would like to set up the process, and sometimes there is really necessary to go high with the voltage. In this case, these high voltage sources have some limits, and it isn't for the long time operation of our devices. We don't want to be still on the limit. This is why it's much better to give each of them some amount and like to say, not to destroy them with the high voltage use at once, and second will have just the zero. So it's also, I think, important.
Thank you. So a very specific question, medical one. Will nanofiber application help in curing the cornea blindness using artificial cornea using drug-eluted nanofiber? Whether any clinical trials, whether any clinical trials was done at your university?
Unfortunately, at our university, we didn't done such clinical trials. On the other hand, from literature, we know that there is a significant effort in this direction.
So how can we assure uniform drug content in particular dimension of nanofiber matrix? Is there any in-process quality test for this? How do you control or check the uniformity of drug release or, let's say, homogeneous content?
We don't have any in-process testing. We of course have to produce the material and then test it from the classical statistical rules from different parts. Of course, we took the material from different parts of the membrane and then we tested the release. Of course, the burst release and sustainable release, and it's really depends on the final application, because if we would like to have some material where should be the sustainable release, we are using materials which aren't water soluble or something like that, and we supposed, or we measured, of course, during these testing how the drug is going outside, let's say, from the fibers, and if it is because of the diffusion or if it is after some degradation started and so on. So it is like that. And but for example, if we are dealing with the special type production of special type of the dental flosses, where everything has to be done in several minutes because nobody will clean the teeth hours. So in this case, it is important to study really the burst release, if all the active agents which are in the nanofibers really play their role, because otherwise it will be waste of the material if it stay inside. So it is like that, but we don't use during the process the in process, let's say, in situ measurements of the uniformity of the drugs, but the post process.
Okay, so next question. What do you think is the best substrate for medical applications, for production of nanofibers for medical application?
A good question. I think that nowadays there are on the market a lot of polymers that should have a significant potential for creation wound healing materials. For instance, Chitosan is one of them. So that is why I believe that there are enormous number of opportunities for the next development in this area.
But I think that the question was, what is the best substrate? It means the supporting material, like a spun bond. Yes, yes. So we are mainly using the non-wovens, which can have also some special treatments, let's say. But the most of our materials is when we tested them or when we distributed them for the other in vivo testing, clinical testing, are without the supporting material. The truth is that the supporting material can really change the morphology. So if the supporting material is, for example, calendarate non-woven, finally, if you check the thickness under the scanning electron microscopy, when you do the cross section, you can see that there are differences according the material.
The structure is copied.
Exactly,
right,
exactly. So sometimes it's good to use a paper if you are sure that there won't be no, I mean, silicone paper, if there isn't, if you are sure that there is no rests, because the surface of nanofibers, as it is huge, it can be very easily contaminated. So mostly we are using spun-bond polypropylene material because we know that no contamination from that material is for our samples, for wound dressings and so on. We had to test it before the clinical study that there is not only the contamination from the supporting material, from the substrate, but also if there aren't, for example, rest of catalyzers for the polymerization of polycaprolactone. We showed all these certificates. So this is like that.
So there are two questions about maximum GSM, like meaning grams per square meter weight we can make using Elmarco technology. So I would say that in my experience, my own experience, I think we made something like 400 grams per square meter, very thick membrane from PVDF. It took a while because a thicker layer you are making, you could start facing some, let's say, high resistivity of the substrate, so the process is less productive, so it takes time. But my experience, my own experience is with the material about 400 grams per square meter. I don't know what's about you.
As I showed the nanofibrous material with these nanoparticles with bismuth oxide, it's extremely heavy. Let's say, what is it? Atomic form, let's say, or particle. And so as Ivan said, if you have only the polymeric fibers, I really would agree with hundreds of grams per square meter. But if and long time of the production, but if I had these composite nanofibers were inside, where this huge amount of the heavy particles, easily and quickly I have 500 grams per square meter. Only there was if I wanted to go up, there was a problem with the separation, that it was too heavy. It a little bit fall down or separated from the support, but definitely hundreds of grams per square meter are easily be done.
Very focused question on our presentation, and one of the questions which was already asked about sterilization and final morphology. Did you study the morphology of fibers of the layers after sterilization?
If again I can, yes, we did a deep study. It is really wonderful question, because it's our, let's say, latest, what we just now actual topic, let's say. Because we aren't using only the polycaprolactone or polyvinyl alcohol, but also some more sensitive copolymers, for example, lactic acid and caprolactone together in copolymer. And it was for one project where we are producing bandages for the blood vessels. Sometimes the doctor said that you needn't to have the bypass, but your blood vessel increasing the diameter in some part. In this case, we are producing nanofibrous bandages, which are a little bit decrease the diameter and help to create new tissue and so on. And there we really tested the gamma irradiation and the ethylene oxide sterilization, and we found for some of these materials during the a little bit higher doses of the gamma irradiation, not the morphological changes too much, but the changes in the internal structure. There the degradation started. The decrease the molecular weight. What we thought, it could be problem because the degradation can be not accelerated, but can be shorter time. It can quickly degrade such material. It wasn't proved by our study, by in vitro studies, but definitely we know that some kinds of sterilization can change mainly the internal structure, because once if you see that it change also the morphology or color or something like that, you can be sure that it isn't good for your material, definitely.
Yeah, maybe I can add, that's an interesting question, because if you prepare something like a perfectly smooth nanofibers, then it is not true that the material will be the same during the healing process if polymer is biodegradable. So it changes. There exists something like a dynamics of the morphology, and some or several such figures Eva shown. So the degradation completely changes the smoothness of the nanofibrous materials.
While talking about healing, about change of dimensions, one of the questions was related to what was the total time of healing during your trials?
So the clinical trial was for just 4 weeks, but the final healing was about 6 or 8 weeks. It depends on the character, no doubt, on the dimension of the wound, and also on the physiological state of the subject.
Yep. Thank you. So also question to the presentation: what type of polymers do you usually use for medical applications, and what type of solvents?
So, as we showed in the presentation, mainly we are using aliphatic polyesters, as it is polycaprolactone, polylactic acid, polyglycolic acid. Also, we are using copolymers. There is huge amount of different types. It means you can use different molecular weights and also different basic ratio in between monomers. So these we used most. We like them because we know how to work with them. But also one colleague is long time dealing with the polyvinyl alcohol, because when we are using aliphatic polyesters, of course, they aren't water soluble. So to incorporate these sensitive active agents which can denaturate is impossible. This is why we are also we work with the polyvinyl alcohol mixture, polyvinyl alcohol, polyethylene oxide, and these polymers can be dissolved in water or the mixture of the water and ethanol. Sometimes it helps decrease of surface tension to have such a huge electrical force. And the truth is that I think that the question is maybe if the solvents are toxic or not. The aliphatic polyesters can be also, I mean, polycaprolactone, for example, or the copolymers can be spun from the carboxylic acids. But in this case, you have to be really careful of the stabilization in a solution. So we also played a lot with it. Professor PhD students did a huge work on it. So now we know that from the carboxylic acid, you are definitely able to electrospun by DC or AC electrospinning these materials, and it works well. So you needn't to use only absolutely toxic solvents as dimethylformamide and so on, but also there is a possibility of, let's say, greener solvents.
Yeah. So question it came to my mind as well during your presentation, a very interesting part, spinning off metal oxide. You mentioned that it's 95% of metal oxide particles, only 5% of carrier polymer. What is the parameters of such solution, meaning like viscosity and conductivity or other important parameters for electrospinning?
So the truth is that I am not able just now tell you real numbers. I have to check to my notes, I'm sorry. But the it was also absolutely surprising for me when I started with it. I had a wonderful colleague from our department of the chemistry. He unfortunately died 2 years ago. And he still pressed me to try more and more of such material into the nanofibers, because the professor said at the beginning, as he is physicist, that as much of the material will be inside, the X-ray shielding will be better, of course. We only have to check the uniformity and so on. So we started with, let's say, 5 weight% and weight%, and still I go up, and it still worked. The truth is that only the trick is that the nanoparticles are distributed from the solvent. It is used by the sonification with ultrasound, and then the polymer is done. And it is fascinating that such huge amount of the particles inside the suspension, if the solution, if the solvent and polymer are chosen right, that the viscosities aren't so huge. That it was for us extremely surprising. Because when I started, I mixed it at the beginning, solvent and the particles just with the glass stick. It was like the toothpaste out. So I said, impossible. But then when I did the sonification, the particles were separated. And when the polymer was done and the mixing at the magnetic mixer, the structure absolutely changed. There was a loss of such, let's say, non-Newtonian behavior as yogurt or something like that. And it really worked.
So actually the mean of the mixing is very important, right?
Definitely.
How you mix it,
Definitely.
mix
The
it with
preparing of the suspension, which is in this case, or solution with particles. I don't know if it is exactly suspension, some mixture. There is a solvent, particles, and polymer is extremely important how we prepare it. But I was surprised how it worked as the nanospider. That nanospider liked it a lot, and the spinning was wonderful there.
So and maybe last edit to it. So that meaning the conductivity of such a prepared solution with the metallic, let's say, nanoparticles, or was not like problematic in electrospinning.
True is that it was oxides. So these particles were metal oxides, which aren't so electrical conductive. It was bismuth oxides. On the other hand, when I also worked, for example, with the ferrite oxides or magnesium oxides, I didn't see so much changes. So it also worked for these parameters, just they don't shield the X-ray. The bismuth was better for the application, so I showed it, but it is possible. And I didn't see if the particles are separated from each other by the sonification. I cannot see problems with the high conductivity, for example, that it would hamper, for example, the spinning.
Okay, thank you.
Thank you. I have to say that our QA section today will blow up soon with the questions. And unfortunately, we are running out of the time. So I will try to say ask 3 questions in one. So please be very concentrated, not quick, but concentrated. So the question is, what is difference observed between quality of medicine nanofibers developed using AC and DC electrospinning? So this is the first part of it. The second part of the question is, it's a quite common question, and now you can see a lot of articles say describing a core shell fibers. So what is your experience with the core shell fibers and in general, and then nanofibers and for medical applications based on AC and DC? What is like?
So fine, I will start the answer about the comparison AC and DC electrospinning, because that's something what we are what we are doing just now intensively. I have to underline that in principle, there is no difference. The structure of AC nanofibrous materials are more tortuous. There are more twins of nanofibers because during the process, positively and negatively charged nanofibers are attracted one to other, or each other. On the other hand, the AC electrospun nanofibers are a little bit more fluffy. And there are some differences in so-called spinabilities. Some polymeric solutions that were tuned for DC electrospinning are not spinnable using AC and vice versa. That's really interesting. I have an opportunity to say something what was missing in our discussion. I have just to underline that also nanofibrous materials for wound healing are excellent because they are enormously flexible. So that is why they are able to copy the relief of the wound. So thank you.
There was also the question for the core shell of coaxial fibers. So maybe Professor should continue because he worked much than me in this topic. What we know that it is only that it's extremely difficult. It's extremely difficult. We used mainly the needle electrospinning for that with special coaxial needles, and set up the process to produce it uniformly was extremely difficult with the flow rates of both core and shell. And on the other hand, what was the interesting problem which solved one, not solved exactly, but was solving one PhD student of professor, was how finally be sure that all the fibers are coaxial, and if they aren't, so how much of them is coaxial, how to prove, because if you are below let's say diameter, which is below the V length, it means, let's say, fibers below 400 nanometers, definitely you cannot use any optical microscopy. So how to be sure? And under the electron microscopes, if it is a transmission or scanning electron microscopy, still you see only 1-2 fibers. What is possible to find that they are coaxial, wonderful. But if you would like to check meters, square meters, it's extremely difficult. So you can do it with some secondary way to put inside some color, and then for example, fluorescent and so on. But I think that it's really the most, it's huge task. And maybe it is bigger than to produce such materials is to be sure that what you are really producing, if it is repeatably, uniformly, and so on.
But more important would be to analyze it.
So, yeah, just I wanted to highlight one part of the question is, what is your feeling about the ratio between nanofibers made using AC and DC for medical application in your group? Just particular example, what do you use more or less?
Nowadays, we are doing both of them. Nowadays, we are doing both of them. Yes, the truth is that we have a project where we have to use the AC because we have to produce there the linear materials. And for this, I mean the hybrid yarns, composite yarns, and so on. And for this is much better AC. So we are using AC. Or if the material should be bulky or fluffy. But if we would like to produce membranes, where we, for example, have the project from the bandages, or where we would like to produce wound dressings, in this case, definitely, we won't use the AC, but we will use DC needleless electrospinning. But the truth is that we still use both of them. And I would say that all days, both of them, still someone is working with something.
I would like to add that both these technologies have something like advantages and disadvantages. In my opinion, DC electrospinning and Elmarco nanospiders are really super to obtain layers of nanofibers. On the other hand, AC has enormous potential to create linear nanofibrous materials, and that is why nowadays we are trying to develop so-called sutures.
Thank you.
Yeah, so just this small add-on from my side on core shell fibers, because we have a particular experience and we know that it's not easy. Like with the needles, I would say that it's relatively clear how to create the setup to make it. Okay, it's not easy to really analyze afterwards. Is it really core shell? Is it a core shell on the whole fiber? length or not. Using Nanospider, it will be more not about the equipment setting, but it should be more about solution formulation. Could be about concentration, but we would say that it's more about adding different mixing or blending different solvents with a high evaporation rate or lower evaporation rate. So this is at least what is our experience about core shell fibers made by nanofiber, by Nanospider. But at the same time, I fully agree that it's not easy then to really understand what is core, what is shell. It's like with the analyzers, you can use, for instance, contact angle evaluation, but it's not usable for all applications. So we really run out of the time, unfortunately, because it was very, very interesting discussion. A lot of very good questions. Thank you very much. So I want to highlight one more time that you can meet us at Medica exhibition in Düsseldorf. Very soon you can ask your questions at this event, and also you can contact us via email or our contact template on our website if you want to really say clear any questions what you had after today's presentation. So thank you very much for your attention, and see you next time. Thank you very much.
Thank you.
Thank you.
Thank you
Thank
too.
you for the organization.
Thank you. Goodbye,
Bye.
everybody.
Bye, everybody.